Click for IB Biology, Photosynthesis. In this video we will be looking at what is known as the light-independent reaction, otherwise known as the dark reaction, in which we will see the cyclic metabolic pathway of the Calvin cycle. We will see carbon fixation under the action of the enzyme Rubisco, sugar formation, a brief consideration of alternative products to be made from the cycle, and the link to biomass and ecological issues. There's a PDF of this presentation in the description below, along with a quiz that you can use to test your understandings. So although it's a cyclic metabolic pathway, we can break the individual steps down into the following. Like all cyclic metabolic pathways, there is what we call the acceptor stage, and the acceptor in this case is the molecule Ribulose Bisphosphate. So that's a 5-carbon sugar with 2 phosphates, and this is going to accept carbon dioxide from the atmosphere, and of course this is what we would call carbon fixation at the biochemical level. The action is controlled by the enzyme Rubisco, Ribulose Bisphosphate carboxylase, a very abundant protein in living things, in plants, and yet, despite its importance in fixing carbon dioxide, it's a relatively slow enzyme with a slow turnover rate. So the addition of the carbon dioxide here would give us a total of 6 carbons and 2 phosphates, but this immediately lyses into what we call GP, or glycerate 3-phosphate, but you can happily abbreviate that to GP, but try to remember that it's a 3-carbon sugar. The next stage is known as the reduction stage, and here we will be using up the reduced coenzyme and ATP, which were generated in the light-dependent reaction. So this is where the light energy transformed into reduced coenzyme and ATP is now being incorporated into the building of biological molecules. GP is converted into a 3-carbon sugar called Triose phosphate. At this point, the cycle can then diverge in 2 directions given by C and D. So if we take the 3-carbon like this, 3-carbon sugar and a phosphate, if we take 2 of those, then we could build a hexose diphosphate. And so now we have come the full length of photosynthesis that we have created a hexose, for instance glucose, which classically is associated with being the product of photosynthesis. Of course, like any cyclic metabolic pathway, it's necessary to regenerate the acceptor molecule Ribulose Bisphosphate, and so we have a regeneration step which is also possible, in which the 3-carbon phosphate is turned into 5-carbon 2 phosphates, which is RuBP, Ribulose Bisphosphate. So this is the regeneration stage. Okay, let's take a look at this in a cyclic diagram. So we have the incoming carbon dioxide fixed by the enzyme Rubisco, fixed with the acceptor molecule of RuBP. The 5-carbon plus the 1 carbon gives us 6 carbons, but that immediately generates G3P here, which is a 3-carbon. So that is okay to call it GP. Then we have the reduction step, in which we go to Triose phosphate, but the important thing is to understand how the energy from photosynthesis is captured by the light-dependent reaction is now being incorporated into biological molecules. At this stage, we can take off molecules like this to generate hexoses. There are pathways which allow us to generate fatty acids, and others still, when combined with a source of nitrogen, to create amino acids. In this way, plants create the biomass that we associate with producers. And of course, we have to have the regeneration of the Ribulose bisphosphate. Now, we know that all chemical reactions need to be balanced, but in analysis, what one would find is that for the turning of the Calvin cycle, approximately 5/6 of Triose phosphate is used to regenerate the acceptor, whereas 1/6 is used to create the hexose. We'll now consider, in the link which is appearing below, the factors affecting the rate of photosynthesis. There's a subscription button for you to click if you choose, and a link back to the playlist so that you can look at the other videos in this series.